Check the description section for time stamps. In this video, I would intensively explain the things that you should understand on nucleic acid and nucleotides. I will start with what? The central dogma of molecular biology. And this is how the video would look like. So I would use that to be able to explain DNA, I would explain mRNA, tRNA, and all other things. This is exactly how we get proteins from what? From our DNA. Now, the next thing we are going to talk about is what? Nucleotides. I will quickly talk about the common examples of nucleotides that we have, like ATP, GTP, CAMP, and the likes. So that's just what you can see right here. The next thing is that we will talk about the basic components of a nucleotide, like the nitrogenous bases, which we can also call nucleobases. We will talk about pentose, that's 5-carbon pentose sugar. We'll talk about phosphate group. Actually, the pyrimidine you can see here is an example of what? Of the nitrogenous bases. So we'll talk about that in details. We'll also talk about the difference between nucleotide and nucleoside. You see, just only T and S is talking about phosphate group. I'm telling you, you need to watch this very video. We'll talk about DNA and RNA, where I would explain what we mean by 5' to 3'. Then I'll quickly talk about the non-covalent structure of our DNA so that we'll understand many things apart from from just the covalent structure that we've been talking about. Now, we'll also talk about the contribution of all these scientists: Chagall, Rosalind Franklin, James Watson, Francis Crick, and all other people that contributed to what we know about DNA. Now, after that, we'll talk about DNA and RNA. I would state the differences between these very two stuffs. We are even going to talk about base pairing. Why do you think that G to C bond is 3 hydrogen, like we have 3 hydrogen bonds? Why A to T, like A to T, that's adenine to thymine, we have what? 2 hydrogen bonds. In this very video, you will learn more about that. I tell you, you must not miss it. So try to fix a time. If you can download it, watch it, and get the best. You can also check the description section for more of those things I've actually aligned there. If you need timestamps so that you will be able to navigate through this very video, you will get the exact parts that you need in this very video. Please don't forget to subscribe to this very channel, because I'll be uploading more videos just like other parts of biochemistry, like when we are talking about when you're talking about lipid, when you're talking about protein and all that, so that you can actually prepare well for your examination. Do me a favor, like this very video. It encourages to do more. Let's start. To understand nucleic acids and nucleotides, I must talk about the central dogma of molecular biology. I'm talking about the central dogma of molecular biology. It is actually how we get protein from DNA. Do you understand? How we get protein from DNA. Now, what do we mean by DNA? DNA is what? Is deoxyribonucleic acid. What is RNA? RNA is what? It's ribonucleic acid. Now, we have different forms of RNA, but I will just talk about the three important forms of RNA. That's the mRNA you can see here. mRNA, that's messenger ribonucleic acid. You can see messenger RNA. Now, we also have transfer RNA, that's tRNA. That's the tRNA you can see here. And the meaning is what? Transfer ribonucleic acid. We also have rRNA, that's what? Ribosomal ribonucleic acid. Now, after we've talked about this, let me quickly explain what we mean by by central dogma of molecular biology. Actually, this is it. From the process called transcription, we can get mRNA from DNA. Do you understand? Now, this mRNA that is being gotten from DNA through the process of translation, this very transfer RNA that I talked about, the transfer RNA you can see here, would help it. It would help it to be transferred to what? To what we mean by the ribosome. You know, actually, when we're talking about ribosome is the what? Is the
organelle that is responsible for protein synthesis in what, in the cell of what, of an animal. Do you understand? Or plant. You should understand what I mean by ribosome, right? It's an organelle that is responsible for what, protein synthesis. Now, this very transfer RNA will transfer that messenger RNA to the what? To the ribosome. And then now, after it has been transferred to the ribosome, there is what we mean by ribosomal RNA in what? In the RNA, in the ribosome. So it is this very ribosomal RNA that what? That would translate all these stuffs and what? And it will be turned to what? To protein. So during this process of translation, we need the help of transfer RNA and the help of ribosomal RNA to make protein from mRNA. You could actually be thinking that why should you call this very stuff dogma? I tell you, the meaning why it is called a central dogma is that what? This is a belief that they don't expect you to question at all. Forget, I've told you, it is central dogma. This is the central dogma of what? Molecular biology. You can get protein from DNA. That's all. Do you understand? So that's the reason why it is actually called central dogma of molecular biology. And as we proceed in this very video, you would understand the difference between DNA and RNA. Many things about that, just like I itemized in the introductory part of this very video. So stay along and watch to the very end. Now, because of this very DNA and RNA, you should see a lot of structures and all that. So with this, we need to actually crack these very structures. We need to break these very nucleic acids down so that we can know, we can get what, our nucleotides from it, and with that we'll be able to what, to talk about how their structures actually inform their what, their functions. So let's go ahead.
What are nucleotides? Nucleotides are the monomeric unit of what? Of nucleic acid. And the example of nucleic acid is what? Is DNA. Now, this very DNA has various nucleotides as what? As its structure. So we have various type of nucleotides coming together to form the structure of what? A DNA. Just like the example you can actually see here. Now, this very DNA, after you've known this, we have examples of common nucleotide also that I would like you to also understand, so that as we go further, as we treat these very nucleotides, it will be familiar to you. We have ATP, and ATP is what? Adenosine triphosphate. And we have GTP also, that's guanosine triphosphate. This is ATP, that's adenosine triphosphate. And we also have the GTP here, that's what? That's guanosine triphosphate. Don't worry, you understand the structure. And that very ATP is what? Is the main energy currency of the cell. GTP also is what? Is an energy currency of the cell. Just like you can see in those structures, ATP and GTP, you should understand that what? We need to deconstruct these very structures so that we can understand it. So now, this very structure of a nucleotide, it what? It has three components. And the first component is what? Is a nitrogenous base. The second one is what? A pentose sugar. And the third one is what? Is a phosphate group. So let me start with what? The pentose sugar. We have two types of pentose sugar in our nucleotide. But let me just start with the pentose sugar. We'll explain that briefly. Uh, this is what? This is the pentose sugar. So, okay, this is the structure of the pentose sugar. Now, we have what? We have OH here. We have this place also. 5 carbon pentose sugar. Some people call it 5 carbon pentose sugar. You just know that the pent there means what? Means 5. So pentose sugar. So this is what? This is ribose. This is ribose. That's what? That's an example of what? Of this very pentose sugar. So let me write it down. Let me write that out. Ribose. That's the first building block. And the second one also is what? We have 2 deoxyribose. So the difference between ribose and deoxyribose is that what? Is that there will be OH and there will be H. The removal of what? Of 0 or of O.
That's just the difference. So let me just or so instead of this very OH, it will be what? It will be H. Do you understand? So if you see something like this very H, that means that what we are talking about OH, that means we are talking about ribose. And then if you see something like H, we are talking about what? Deoxyribose. And I tell you, these two difference, it is only a difference that we can actually talk about right now. It states the difference between DNA and what? And RNA. It states the stability and all that you can actually see in DNA, and you can't actually see in what? In RNA. So these two deoxyribose is found, the pentose sugar that is called 2 deoxyribose is found only in the what? In our what? In our DNA. Do you understand? So note that. Why ribose is found only in the what? In RNA. Now let me quickly number this very what? This very pentose sugar. We have 1 here, we have 2 here, that's our 2. We have 3 here, we have 4 here, we have 5 here. Now to differentiate this very numbering system from what? From another ring, because we have two kind of heterocyclic compound in what? In our nucleotide out of the three. That's the base and the pentose sugar, we are still going to talk about nitrogenous base, where I would teach you how to understand the structure. Don't worry about that. But in this very place, I just want you to understand that what? For us to differentiate um this very uh this very this very pentose sugar from that very base, so that you can understand. And even while talking about that very structure, we have to what? We have to add prime to it. So that will be what? 1 prime, you have 2 prime, you have 3 prime, you have 4 prime, and you also have 5 prime. So that's how we number, what would I call it, our pentose. And with this, you should understand that this very 2 deoxyribose also should be what? 2 prime, because as that very 2 prime, we have a deoxidation. So our what? OH is being deoxidized to what? To H. Now this is just the rough sketch of what we mean by nucleotide. So we have our phosphate group here, we have our what? We have our ribose or deoxyribose, that's our sugar here, and we also have our what? Our base here. So we've talked about this very what? We've talked about this very sugar. Do you understand? So now what we'll do now is what? To talk about the phosphate. So let's continue with the very phosphate you can see here. So this is the structure of a phosphate. You have P being attached to O. Now we have O here also. That's it has minus, and there's a double bond here. Do you understand? So it could be monophosphate, it could be triphosphate, it could be diphosphate. Do you understand? So with this, you should be able to construct triphosphate, monophosphate, and all that. So when we're actually talking about a nucleotide, what you would see, you will realize this very structure as what? As phosphoesters. Do you understand? So let me just write that out. Phosphoesters. Phosphoesters. I'm sorry. Phosphoesters. Phospho Phosphoesters. But when we're actually talking about what? The structure of our DNA. DNA is a double-stranded helix, and what? We actually see phosphates occurring as what? As phosphodiesters. It's actually used to what? To combine all these nucleotides together. You understand? So let's just write that out. Phospho phospho In the DNA, it's occurring as what? Phosphodiesters. Diesters. So roughly you should be able to know that what? This very bond, we have 1, 2, 3, 4, 5, 5 bonds attached to what? Attached to the phosphorus there. So that you just understand this very thing, and you should see that what there is a negative charge at what on this very O that is what beneath that very phosphate. So that's just that very phosphate group. So let's move to, now that we've talked about phosphate, we've talked about the sugar, let's talk about the what? Let's talk about the base. Now, while talking about the base, we have two types of these very base. We have what? We have number one, we have purine, and number two, we have what? Pyrimidine. Do you understand? So let's start with purine. Now, we also have two types of this very purine. That's what? Adenine and what? I'm sorry, adenine and guanosine. So I'm going to draw adenine here around this side. So just note that. And I would have what? I would have guanine also at this very side. If you do know how to draw this very structure, you can just skip this very section to the other part. You can check the description section for the timestamps. Do you understand? Now, to draw adenine, you have N here, and you also have what? You also have N here. So just follow me as you would understand this very structure right now. It's actually a what? It's actually a 9-membered ring. A 9-membered ring. So you have N also here. You have N here. And at the same time, you have N here. So this is the structure of what? Adenine. So just like this also, you would this is not the structure of adenine. No. We've not completed the very structure, but just note that it's kind of like this. So let's do the same thing also with guanine. Let's do the same thing also like this. Let's draw the same structure in this very guanine, so you see the difference right now. So after you've seen this, now the difference between adenine and guanine, we have a double bond here. You have a double bond here. You have a double bond here. A double bond is here. You understand? You have a double bond here. That's for adenine. You also have a double bond here, and we also have a double bond here, making 4 double bonds in what? In adenine. Do you understand? So, and to cap everything up, we have NH2 at this very position. Do you understand? Now, this very place, that this very n, we have what? We have this very nitrogenous base attached to what? To the what? The sugar. You know, I was talking about pentose sugar when I was explaining ribose and 2 prime deoxyribose the other time. Now, it is at this very end that the what? The what? This very base will be attached to this very sugar, just like in this very position. Let me show you this very position in that diagram I drew the other time. You understand? So that's exactly where. So let me now draw the okay, let me talk about guanine also. This is guanine, right? So this very guanine, we have a double bond here, a double bond here. There's a double bond here. Would have it would have a double bond here also. And at this very side, at the top here, you would have a what? You have O, that's oxygen, and you would have NH2 here. You have NH2 here, and you would have H here. So Do you understand? Now it will be also attached to what? To the sugar at this very at this very end at this very end. So we don't really know the end now, right? We don't know the number of that very end. So let me quickly tell you how we note how we the numbering system in the what in the purine. We actually have this. This is one. We have this to be one. We have this to be one. We have this side to be two. We have this side to be three. You understand? We have this side also to be four. We have this side to be four. We have this side to be five. We have this side to be six. Now, talking about this place also, you have this place to be seven, eight, and nine. So, how we actually number this very thing is at first you will start with what? An anti-clockwise numbering. That's what you will number it like this. That's how the arrow would be. Then you would number it back like this. That would be what? That would be the clockwise direction. Do you understand? So that's the so with this you should actually know that what this very purine is being attached to the what to the sugar at what at N9 position. You can be asked in your exam. Do you understand? So just note that it is being attached at the what at the N9 position. So just note that N9 position. So any purine, just like this one also, if you number it right now, you would also get this place to be what? To be 9. You understand? So all purines are what? Are number are what? Are attached to the what? Attached to the sugar, the ribose or the deoxyribose at what? N9 position. And there's another thing I would like you to understand. This would really help us to understand nitrogenous base. Do you understand? We can also call that nucleobases. So let's just write the second type of what nitrogenous base, that's pyrimidine. After that first one, the first one is purine, right? So the second one is pyrimidine. Uh okay, let me underline that. So with this, you should be able to understand purine and pyrimidine. You will not be able to you will not what you will not give the structures of purine to pyrimidine, and you will not give purine the structures of pyrimidine to purine, if you can understand this. And it is very simple. Small name, big structure. Big name, small structure. If you check this very purine, you see that what it is not lengthy. It has a small length. Why this very purine pyrimidine has what has big length. So that's just it. That's with this you should be able to know that what purine would have a big structure because it has a small length. So that's why it has 9 membered ring. Actually, it's a logic for you to understand it. It's not the main reason why you can actually see that what the ring there should be 9. No. Now we're talking about purine also. Purine is what is it has 6 membered ring instead of 9. It has pyrimidine rather. Pyrimidine has 6 membered ring instead of what 9 membered ring. So it is actually a logic coined by people so that you can understand this very nitrogenous base. So small structure, big big name, and big name, small structure. Just like I said, pyrimidine is a what? Is a 6 membered ring. Do you understand? So we have pyrimidine has 3 basic structures that we can actually see in the what? In the nucleotide. That's uracil, we have cytosine, and we also have what? We also have thymine. Now, these very three, we have one that is in these very three, we have two present in the DNA, we have another two present in what? In RNA. So none of these three is at the same time present in the DNA and the RNA. Do you understand? No. So let me quickly talk about the three structures. The first one I would draw here will be what I will talk about here will be what will be cytosine. I expect you to know about cytosine sef. And I will also talk about uracil. So I will draw uracil here. And the last one I will draw would be thymine. So let me quickly write thymine here. And don't worry, you would understand these very concepts very well. Timing. Now, after we've known this, let's commence with the drawing of this very cytosine. So we have N here, and we also have N around this very side. So it's just a six-membered ring. A six-membered ring. So let me do the same thing with uracil also. Uracil, don't worry. So just let's be very fast with that. And a word of timing. So let's draw that also for timing. So we are done with that. And now let's quickly talk about where you would find the double bond. Yeah, in timing you will find a double bond here. Also in uracil you will see a double bond here, and also cytosine you will see a double bond here. Now there is another thing that is being added to what to a cytosine. There is another double bond around this very side. So cytosine has two double bonds. Why this very uracil and thymine you will not see another double bond there, just only one. So that's just it. And we are not done. Cytosine, so in addition we also have what it is NH2 is attached to this very cytosine and this very side, just like that very adenine. If you remember, we have what we have this NH2 attached to the what to the carbon atom. I said the carbon atom. I'm sorry. That's very around this very side, around one ring there, and we have also another one here. That's the double bond O. So with this, we are done with the structure of what cytosine. So let's talk about the uracil as well. Uracil is what we also we actually have the difference between. Okay, I'll still talk about the difference between uracil and thymine, but just note that what this very uracil, it has what it has O here, and instead of NH2 here, it would have O here as well. Do you understand? And the same thing also with thymine. You will also see something like that, just like that here, and at the same time you see O here. Now, what is now the difference between these very two structures? Actually, here we have H. That's for uracil, and in this very place you have what you have methyl. Instead of H, there will be what there will be methyl there, and that is the difference between what between thymine and what and uracil till today. So you can actually find cytosine in what in both DNA and RNA. So let me write that out. DNA. You can be asked all these questions, so just try to take notes, or if it is for knowledge sake also, try to understand this very concept. So you can find that in what both DNA and RNA. Uracil also, it's only found in what in RNA, and why thymine it's found also in what in DNA on norms. Do you understand? So it should be found in DNA, and also uracil also on our norms should be found in what RNA. So let's talk about the numbering system um, of this very nitrogenous base, that's purine. The numbering system here, just like what I talked about the other time, we have we have um, we have this place. Okay, this one is different because instead of this is actually where we name our ours to be what our one. So that will be what that will be one. Then we'll do it. We'll go through two, 3, 4, 5, then 6. So you start from this very N1 at this very place. So N1 position. And just like that also, you can also see that this place will be 1, this place will be 2, this place will be 3, this place will be 4, this place will be 5, and this will be 6. So just note that uh in this very type of, in this cytosine, uracil and thymine, you have it, you have this very non-brain system in a what, in a clockwise direction. Clockwise direction. Why in the purine you start with a clockwise direction, and while talking about the second ring, you would what, I'm sorry, in the purine it will be what, it will be in an anti-clockwise clockwise direction, and after you are done with the first ring, the second ring will be what, will be in a clockwise direction. So that's just the difference, you understand. And note that what, it will be attached to the what, to the ribose or deoxyribose. That's the pentose sugar at what, N1 position. Is a typical exam question that what, that you could just be asked. So this place will be what, will be, it could be the ribose, this place could be the ribose, or it could also be the what, the deoxyribose. Just note that what, it is being attached to the sugar as that position N1. So it's another thing you should jot down, and hope you have a jotter there sef that you are actually jotting this very thing, or you just, no, please try to jot those things you learn here down. The faintest ink is better than the sharpest brain. Now let me quickly take you back to the examples of nucleotides that we treated the other time. Now I told you that what, I told you that ATP is an example of a common nucleotide. Now look at this very side, just like what I taught you now. You can see that what, this is actually what we mean by what, our sugar. Do you understand? That's our sugar, and that's it. And this is our what, this is our phosphate group. This is our phosphate group. We have our phosphate group here, and the other one you can see here is our what, that's our what, our nitrogenous base. That's all. Similar to that also, we have this also as our nitrogenous base. We have this as our what, we have this as our what, as our uh pentose sugar, and we also have this very place as our what, we have this as our what, our phosphate. I tell you, you are done. So that's just it. All this very structure that looks complicated, you see that what, little by little, all these structures that look complicated, you see little by little, you can actually see that what, it's a very simple thing. And the reason why it is triphosphate, if you check this very place, you see that what, you have actually 3 phosphate here. We have 3 phosphate here. So we have 1 here, 2, 3. So that's why it is triphosphate. 3. And that very adenosine that you can see here signifies adenine. If you check that very structure very well, if you check the structure of that very nitrogenous base, you see that what, it's actually a structure of what, of, it's actually a structure of what, adenine. That's one of the nitrogenous bases I drew the other time. Now, we also need to look at the structures of these very what, of these very nucleobases. That's what it's called, nitrogenous bases, so that we can actually understand things while I will be explaining the non-covalent parts of our DNA. Do you understand? We need to know where the donors are and the what, and the acceptors are. As you can see, this is what, an hydrogen or what, on a nitrogen. That's adenine, right? You will see that what, that will be a donor. So this place, there's a donor here. On this very place also, you see that what, that's what, this is N, right? And if you check the bonds there, you will see that what, it is what, an acceptor. And this very N also, you see that what, is an acceptor, just like that. Now if you should move to guanine also, you see that what, this very side, we have what, we have um this to be an acceptor. This very side also, we have that to also be the what, the donor. This also to be the acceptor. Because I said acceptor, it should be a donor because
if you actually split this very H2, that's H and the H. So H on a nitrogen would actually give you a what? A donor. So with this, you should understand, just understand this very part, because while we are moving, you should be able to understand, you should be able to relate them. Honestly, looking at these words, I expect it to be like, kilo shele. What's really happening? There is T here and there is S here. So what's not the meaning of that? T and there is what? S here. Actually, let me tell you, the difference between that very T and S is phosphate. I'm telling you, the meaning is this. Let me tell you. Let me actually draw a structure of what, of a nucleoside. The very structure you can see here. So let me quickly draw the structure of a nucleoside. You have you have the pentose sugar I drew the other time. So that's it. This is it. Yeah. So with that, it is being attached. It is attached to what? Let's just it could be adenine, it could be guanine, it could be cytosine, it could be uracil, it could be what? It could be thymine. So let's just write something like nitrogenous base. Do you understand? Just put B there. So it is attached to B. Do you understand? Now at this very side, I told you the other time that what, it will be attached to phosphate, but I tell you, when I'm talking about that very nucleoside, it won't be attached to what, a phosphate group, and that is the difference between that nucleotide and nucleoside. So it is a typical test question. You could just be asked that what is actually the difference between a nucleotide and a nucleoside side. The difference is that a nucleoside is a nucleotide without phosphate. That's just it. Do you understand? So this very T and that S has actually what, uh, removed the very phosphate. Okay, just for example, this very ATP, there is an example of this ATP. For example, ATP, this very ATP you can see here is an example of what? Is an example of what? Of nucleotide, because it has what? It has what we mean by phosphate. This very phosphate you can see here. But without that very phosphate, it is called what? Nucleoside. Thank you. God bless you. God bless you. God bless you. Now, there is another thing I intentionally did not talk about the other time. So that is what? The bond here. And we call that bond glycosidic bond. So let me write that out. Glyco glycosidic bond. It's actually a bond to what? To carbohydrates. But in this very arena, let me just explain it. It is actually formed with what? It is formed in this very junction with what? The removal of H. There's an hydrogen over on this very end. On this very end, you can see here, there should be an hydrogen there. Hydrogen there. If it has not what? If it is not bonded to what? If it is not bonded to this very pentose sugar, do you understand? There should be H there. So this very H will be eliminated while forming this bond, and at the same time there should be an OH on this very what, on this very pentose. There should be an OH on this very pentose. So let me just quickly see how draw how it should be. This should be a pentose, right? It should be a pentose sugar. So you have what? You have this, you have OH here. So there will be what? The elimination of this very OH you can see here. And normally there should be in this very ring. I'll just draw part of the ring, sha. I'll just draw part of the ring. There should be what? There should be H here. There should be H here around this very side. So this very H also will be eliminated. So this very H would combine with this very OH in a reaction called condensation reaction to what? To give out oxygen. I said oxygen, I'm sorry. To give out water, that's H2O. Oxygen, to give out water, that's H2O. Then this very bond would be there. So that would be glycosidic bond. So now let me remind you what I said the other time. If that very nitrogenous base is a purine base,
it will be what? It will be this bond would form at what? At N9 N9 position. Do you understand? And if it is what? If it is a what? Pyrimidine, it will form at what? N1. So exactly. No, I was telling you that time that I shouldn't forget this. N1, it will form at N1. And that's for nitrogenous base, right? So I want to talk about pentose sugar right now. So this pentose sugar will be formed at what? At N1. At C1. I'm sorry, C1. So that will be that will be what? That will be C1. But that very one will be what? C1 prime. Don't forget. I told you the other time that to differentiate the numbering system in the pentose from what? From the nitrogenous base, we have to what? We have to include prime. And I tell you, this glycosidic bond is important, especially when we have to cut out our sugar from our genus base when there is mutation in the DNA, so that it will not what it will not get misread. Why was in the synthesis of our mRNA from our DNA. So we have to just cut that sugar out by that was glycosidic bond. I expect you to understand that this very structure you can see is what? It's just a rough sketch of what would I call it? A nucleotide. Do you understand? And I already explained what we mean by nucleoside. That S that is there includes that what it does not have a phosphate. So let me quickly talk about the naming of nucleotide and what and nucleoside. And with that, let me just take a base. Which of the bases should I take? Okay, let me take guanine. Let me use guanine. Do you understand? So assuming this very base you can see here is what? Is guanine. Is guanine. That's Guanin. That's what guanine is actually what? Is actually a purine. Do you understand? So it has 9 membered ring. Don't forget. You can easily refer back to those diagram, the sketch I did the other time when I was explaining guanine, adenine, cytosine, thymine, and uracil, so that you would understand the structure. It's very very important if your school actually strikes balance and all that on it. But there are some schools that will not actually need you to understand the structure because it is somehow complicated to them. While some schools will expect you to understand it so that at least even in the exam you'll be able to what? You'll be able to draw the structure very very well. Now, using guanine as an example, now let me just talk about the nucleoside of what? Of guanine, just like it looks without the addition of any other methyl and all that. So don't forget that what nucleoside is what is actually the remover of what? Of night of it involves the removal of what? Of phosphoric phosphate group. Do you understand? So that's nucleoside. So the nucleoside now, if you want to name it, and I'm talking about in RNA. In RNA, the guanine nucleoside will be what? In RNA, the guanine nucleoside will be what? Will be guanoside. Guanosine. Guanosine rather. Guanosine. I'm talking about in DNA. In DNA, it would be what? That's double-stranded helix that includes a double strand and all that. You have what? You have deoxy guanosine. And I expect you to know that what the difference between these very two, RNA and DNA, is actually their building block of what? Of sugar. Here you will see that what it is very RNA. The building block will be what? The building block will be tell me now, what is the building block? It's actually ribose, just like I told you the other time. And what is that very ribose? That's the what? The usage of OH instead of what? Instead of this very H you can see here. Do you understand? Now, when I'm talking about this very DNA, the building block or the sugar building block is what? Is 2 prime deoxyribose. 2 prime deoxyribose.
deoxyribose, ribose. And that is what, that is actually the usage of what, that's the usage of this very what, of this very H instead of what, instead of OH. You understand? So that's just the reason why it is called deoxyribose. So if you actually see something like that without the usage of H, if the sugar has OH, that means that what is a what is actually a what a deoxyguanosine. If you see something like this, and don't forget, this very nucleoside means that what, there is no phosphate. So this very structure I'm drawing right now is actually just like this. Yeah, this is it. So where this very place dictates if it will be a ribose or what, or a deoxyribose. You understand? So if you check this very side, you see that what, there's nothing like this very pd again. You can't actually see this very phosphate there again. There is no phosphate here at all. Do you understand? No phosphate is attached to that very guanoside. So just like that, if you have adenine also, we call it adenosine deoxyadenosine. If you have pyrimidine and all that, so you still talk about that in this very video. Now if you should talk about the what, the nucleotide of this very guanosine, guanine, I'm sorry. So the nucleotide instead of the nucleoside, nucleoside is what, is the removal, it includes the removal of what, of phosphate group. Why this very nucleotide is the what, there will be an addition of this very phosphate group, just like you can see here. So if you use guanine in RNA, in RNA, I'm talking about RNA, where we use what, where we use the building block of what, that we use the building block, which is what, which is ribose, the sugar building block, which is ribose. We have what, we have the naming the naming, like the naming of that, the the name of that very nucleotide to be what. So let me just write it down. Instead of guanosine that that you see as the form of RNA in what, in nucleoside, it will be what, it will be guanylate. To be guanylate. So but if it's a, if it's what, if it's a DNA, if it's a DNA, you understand, we have what, we have deoxy. So just note that anytime you see something like a DNA or something like that, what you will just use to differentiate it is what, is deoxy. You understand? Deoxy guanylate, 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 deoxy guanylate. So that's in DNA. And don't forget that here, if it is an RNA, if it is a RNA, this very OH, it will be the user of this very OH, that's ribose. If it's a DNA, the usage of what, of this H, that's what, that's deoxyribose. So in summary, if you have something like adenine, and that's what you can actually see here, and what, and you are looking for the nucleoside. If it is actually in the what, in the RNA, it will be like this. That will be adenosine, and in nucleotide it will be what, adenylate. So that's what. So let me just point it here that what, RNA, RNA. Now this second one also, deoxyadenosine. If it is actually in the what, in the nucleoside, it will be that, and in the nucleotide it will be what, deoxyadenylate. So that's what, that's for DNA. That's for what, that's for DNA. And we've actually treated guanine. So just cytosine also, you can what, you can also see that what, in RNA it will be cytidine, which is an RNA like ribose. If you have ribose there instead of deoxyribose, and if you have deoxyribose, you would have this very name, that very name, you have that very name. You see cytidinylate for what, for nucleotide, and deoxycytidine. And what will you name thymine also if it is a nucleoside? That will be what, that will be thymidine. And I expect you to know that we only find thymine in what,
in DNA. So the thymidine and the deoxythymidine is used for that very nucleoside. And nucleotide also, we use thymidylate or deoxythymidylate. The reason why we use thymidine or deoxythymidine, because normally if you check the sequence, just like what I've been saying the other time, since you should understand that thymidine should be should be for RNA, and the deoxythymidine should be for DNA. But you know that thymidine can't be found in the RNA. Do you understand? Now, to make what we actually pronounce simpler, we can actually say, instead of this very deoxythymidine, we can say thymidine. Do you understand? So it is used interchangeably when it occurs in what, in the DNA. So let me just quickly summarize this very stuff, and I would pick out some of the things that you should understand in this very point before we identify and name some nucleotides. Don't worry, it will be fun. So nucleotides and what, and nucleotide. Let's quickly talk about this. So if you see something like guanosine and guanylate, is what is actually seen in what, in RNA. That deoxyguanosine and guanylate, we actually find that in the DNA. How about cytidine? Cytidine is found and is cytidylate in what, in RNA. What of deoxycytidylate? That's DNA. That's DNA. And don't forget what I said the other time. So thymine will be what? Thymine will be only for DNA. And this uridine and uridylate, that will be seen in what, in RNA. So just note that the nitrogenous bases that could actually be seen in a DNA, we have, okay, let me use the color. So in a DNA, we have what, we have thymidine. No, that's number one. We have what again? We have cytosine. We have what again? We have guanine. We have what again? We have adenine. So that those are the four nitrogenous bases you can find in the what, in the DNA. Now talking about the what, the RNA, you can find what, you can find uracil, cytosine, guanine, and what, and adenine. Now let me quickly say this. Um we're talking about adenine and guanine. They are both purine. We are talking about cytosine and thymine, and uracil. We have them as what, pyrimidine. So let's move forward to name some nucleotides and the nucleoside. You should know that this is a sugar. Do you understand? And this is our nitrogenous base. And if you check this very place, you see that what, there is nothing like a phosphate group there. So that is telling us that what, this is a nucleoside. And if you check the table I drew the other time, you should see that what, the answer to this very question should be what, uridine. So the answer is what, uridine. So that's the name of that very nucleoside. And also thymidine. If you check this very place, you see that what, we have thymine as the what, as the nitrogenous base, and there is nothing like phosphate here, so it shows it is a nucleoside, and the name given to it should be what, thymidine. So with this, actually, this very thymine, I expect you to understand how to draw it, to know how to draw it. You will not write thymine if you were actually asked in the exam. You shouldn't write something like this. You have to what? You have to draw the real structure of that thymine. Okay, let me quickly draw thymine for you. This is thymine. We have N here, N here. Now you have what? You have though I drew it the other time, but let me quickly just, so that you see this is actually what you should draw there. Do you understand? So this is it. Thymine. And don't forget what I said the other time, that what, there is a methyl group here, thymine. Why in uracil there's what, there's an hydrogen there instead of a methyl group. And here we have what, we have O, and we have what, we have O here. So this very place, at this very n1, as I told you the other time, at this very n1, it will be attached to the what, to the thymidine. So at this very point, so it will be attached at these very points you can see here.
be attached to what? To the sugar. You understand? So that's just this very place will be attached to it. And don't forget it will attach to this very place, this very side rather. We still have a long way to go, so let's quickly talk about nucleotide. Now let's move to naming nucleotides. Actually, if you check this very diagram, you should know that what the presence of adenine and this very what this very deoxyribose as the sugar and what and phosphate group that you can see here signifies that what it's actually what deoxyadenylate. Do you understand? And you should understand that what this is actually what we mean by that very adenine and the other one. So if I should actually change this very H to what? To OH. If I change this very H to OH, if I should add OH here instead of the O that you have there before, instead of the H that you have there before, it turns to what? It turns to what? Ribose. And if you actually have ribose, you should understand that what the name would change to what? To will change to adenylate. Instead of this very deoxyadenylate, you have what? You have just only adenylate. Now that's not what I would actually like to talk about. There's another name we can actually name this very nucleotide instead of what? Deoxyadenylate. That's what we are going to talk about right now. We'll start with the name it should have if what? If it were to be and what? A nucleoside instead of a nucleotide. So that means that what? We'll start with just only naming this very part. You understand? So let's just write that part out. So what's the name of that very part? That if you check that very table, you can go back and check it. You understand? You would have what? You have your answer to be adenosine adenosine adenosine. Do you understand? So after we've written adenosine, what is the next thing? Now this is where the numbering I told you the other time would come in. It comes in here. We have what? We have 1 here, that's 1 prime. We have 2 prime here. Don't do you must not mistake it to be 2. No, it is what? 2 prime. We have what? 3 prime here, 3 prime here, and we also have 4 prime here. We have what? We have 5 prime here. Now the what? This very phosphate group, if you check it, is attached to what? To the sugar, to this very deoxyribose at what? At 5 prime position. So you are going to write what? You are going to write 5 adenosine 5 prime. Now you would write what? So what is the name of this very phosphate group? Is it triphosphate? Is it diphosphate? Is this a cyclic phosphate? If you check it, you will see that what is actually what it's actually a monophosphate, just one phosphate. Mono means one, di means two, tri means three, and all that. So let's just write that very the name there. So it will be what? It will be it will be what? It will be monophosphate. So just write mono for phosphate or phosphate. So that will be adenosine 5 monophosphate, 5 prime monophosphate. So that's just the name. Now we have to draw this second structure ourselves. I intentionally left it like this. I don't want us to what? To draw it before we actually talk about it. So I will need you to draw it in your notes even before you what? Check this very video. You can just pause this very video and draw it in your note, and after that just come back and see how I will do it. I will draw it here. So we have what? We have our pentose sugar. So here would have what? Would have our adenine. Our adenine, if you understand that this will be N. Okay. So here you have it like this. Okay. So you have N here, you have N here, and you have what? You have I'm sorry, shouldn't be like this. You have N here, like this. Do you understand? So and we'll put the double bond here, put a double bond here, put a double bond here, a double bond here, and a double bond here. So don't forget NH that will be here, NH2 that will be here, NH2. So after that, the next thing you should understand is what we are talking about monophosphate. Do you understand? And it's what? It's at the 2' position. So let's quickly talk about the prime position again. The numbering system in the ribose again. We have 1', we have 2'. So at this very position, that's where you would have what? You would have your what? Your monophosphate. So now let's write let's draw a monophosphate there. So it would not you would have O here, and with that it will be joined to what? To other parts of the what? Of the phosphate group. You have minus O minus O minus, then you have what? You have double bond here. Do you understand? So that's just the okay. Let me quickly finish it. We have OH here. You have don't forget this very place where we have OH. So now we are done with what? With the diagram of the of adenosine 2' monophosphate. Now you can actually try to what? To draw adenosine 3' monophosphate also by what? By uh, putting this very phosphoric phosphate group at the what? At the 3' here. Do you understand? That's just it. Oh my god, this thing is getting interesting. I'm telling you, you understand. You see that what? After this, you will talk about ATP that I did show you the other time. So with that very ATP, with that very ATP, we'll be able to draw the diagram and all that, and you'll be able to see the what? The main energy currency of the cell. Just how the what? How the structure looks like, and you'll be proud of yourself that you can actually draw that very diagram. I congratulate you in advance. Now, let's quickly draw what? This very adenosine 5' monophosphate. We have O here. Okay. Yes. After that, we have what? We have our adenine here. Oh, N N. Okay. Okay. Uh. Okay. Yeah, we are going. We are going. There will be N here. There will be N here. So after we've actually done that, what is the next thing? The next thing is for you to put what? Our double bond here, our double bond here, and our double bond at this very side. So put what? Put NH2 here. Don't forget NH2. So with that, the next thing we should do is what? Is to include our OH here, because it's a ribose. If you check, it is what? It's not actually the adenosine. Do you understand? It is actually adenosine. Do you understand? So it will what? There will be OH here also. Now, where is the 5 prime 5 prime carbon? 5 prime carbon. This is actually okay. We'll get I'm sorry, this should be 1. This should be 2 prime. This should be 3 prime, and this should be 4 prime. Why we have okay, I didn't extend this. So there should be something like this here. So that will be what? That will be our 5. That will be our where our 5 prime will be. So there will be what? There will be O here. There will be P here. P here, and there will be another O here. Now we have what? O with a double bond here, and we also have our O here. So there will be what? There will be minus, and there will be another minus here. So we're actually done with adenosine 5 monophosphate. Now, where is the what? ATP I promised. Now, just like I told you, we have what? We have what we mean by ATP here. That's adenosine triphosphate. And some people could also call it adenosine 5' triphosphate, because it is connected to what? To 5' here. Do you understand? So that's why it is what? It could be called 5' triphosphate. And this very ATP, oh, there should be something here. There should be a double bond O here and the minus here. So this very ATP, if you check it, the difference of this very ATP with what? Adenosine 5' monophosphate is just the what? It's just the phosphate group here. If you check the other structure, the other structures, you can see here, both the both the nuclear base, or what we mean by the nitrogenous base, and the what the pentose sugar, and even this very one, you see that what we have the same thing there. It's just that what the phosphate group, the phosphate group is what this one is mono, why this is what, why that is the second one is tri. You see, that one is tri. So, and tri means three, mono means one. So this very that means that what we have one phosphate group here, why here we have what, we have three phosphate group. So this is one, this is two, this is three. Do you understand? So that's the three phosphate group that we have there, and that makes it adenosine 5 prime triphosphate, or adenosine triphosphate, which is what shortened to be our ATP, and it is what, it is actually useful for what, as a form of energy currency in the cell. There's still a lot to talk about, so if you'd like to fast forward this very video, check the timestamps in the description section. Now let's quickly draw what we mean by what this very adenosine 2 prime 3 prime cyclic monophosphate. So as we used to do, the first thing we are going to talk about is what, our pentose sugar, and this is it. So our pentose sugar, we have what, we have that here. So after we've done that, it is adenosine, right? Adenosine, right? So that means that what it will, you would have what, you have adenine here, you have adenine here to be connected to what, adenine. So let's just write this is the structure of what, of adenine. So I'll quickly do that. So after that, yes, there will be a double bond here. Yes, 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 yes. Okay, there should be another N here. There should be another N here. Yes. So there will be what? There will be another one here. Yes. So I will have another N here, and after that I would what? I would have these to complete it. So double bond will be here. There will be a double bond here. There will be a double bond here. There will be a double bond here. So don't forget, in adenine we have 4 double bonds. So after that, don't forget that there should be what, NH2 here, NH2, and after that you are done with the structure of adenine. Now the next thing is what, is that very cyclic monophosphate. If you check that, if you actually see it, you see that what, it tells us that what, it's not actually just a monophosphate, it's a cyclic monophosphate, and you can see that what, it is on 2 prime and 3 prime. So let's get the position 2 prime and 3 prime. We have position 1 prime here, we have 2 prime here, we also have 3 prime here. So it will be on that very 2 and 3 prime. But quickly, let's finish the structure of this very pentose sugar. So there will be what, there will be an OH here. Do you understand? Now the structure of that very phosphate sugar. So after we've done that, we have a monophosphate, right? So what we just do is to what, is to draw, is to what, is to extend this very thing, extend it, then put this is part of the phosphate, and this is another O from what, that's another O from what, from that very phosphate group. Then we have just like you can see that what, it's a cyclic phosphate cyclic stuff. So that's how it would look. Then you put our P at the middle. Your P will be what, at the very middle. So there will be P here. Do you understand? So the what, there will be a side with a double bond, and there will be another side with what, there will be another side with what, with just O. You understand? O. So that will be O minus. So this place will be O. You understand? So that's just the structure of that very what, that very adenine 2 prime 3 prime cyclic monophosphate, and we are done. Now let's look at the covalent backbone of nucleic acid, where we have what, the RNA, we have what, DNA, as what? As the examples of what? Of nucleic acids. Now, talking about DNA, you should know that what? This is actually what we mean by that very DNA. Like I want to use that very structure as an example of what? Of DNA, and I want to use this very one also as an example of what? Of RNA. Now, for us to understand this very thing, I didn't I didn't put the basis there. So I would put the basis there now so that you understand the structure of DNA and RNA. Actually, the basis, we are talking about the basis of what? Of DNA. The basis of DNA, you should still understand that what we have, the first one to be adenine. You have the second one also to be what? To be guanine. You have cytosine there, and we have what? We have thymine. Do you understand? So those are the four bases you can actually get in what? In a DNA, in a DNA. But talking about RNA, RNA is a bit different. You have what? You have the first one to be something like adenine, second one guanine, third one we have cytosine. It's present in the two of these like both RNA and DNA. We have adenine, guanine, and cytosine. But the difference now is the what? Is the presence of uracil here. Why in DNA we have what? Thymine. So now let me just name this very place. You should know that what? This is our sugar. You should know that this is our sugar again, just like that. So I don't really need to add that. So the first base I would talk about right now is what? Is E. Let me that's adenine. Let me use adenine here. The second one also, uh let me use thymine. You know, thymine is also present in the what? In DNA. And the last one, let me just call it guanine. Do you understand? And that's for our DNA, right? So let's talk about RNA right now. RNA, let me just add for an RNA. Let me add. You know, this is sugar. Let me just put uracil here, because uracil is also only in our DNA. I'm talking about this very RNA also. Let me say this is guanine. This is guanine. When I'm talking about this place also, let me say this one also is what? Is cytosine. If you check, if you check all these stuffs, you realize that what? It actually tallies with the word DNA, and it tallies with the word RNA. You know, don't forget, DNA is what? Deoxyribonucleic acid, and RNA is what? Ribonucleic acid. And we have this very phosphate group you can see here as what? As phosphodiester. Do you understand? And just like I told you the other time, let me remind you again. We have phosphate group occurring as what? As phosphoester in nucleotide. But you know, it is we have a lot of nucleotides coming together to form what? To form nucleic acids, right? Nucleic acid. Now, this very nucleic acid has what? It has phosphate group occurring as phosphodiester. You understand? So let me write that out. Phosphoester or phosphodiester. Phospho. So in this very place, you have it as what? Phosphodiester. So when we're talking about nucleotide, you have to just remove this very word di. Do you understand? So that's just it. That's just the difference. So that's what you can actually see here. We have phosphodiester here. You have phosphodiester here. You have another phosphodiester here in RNA, in DNA. Do you understand? Now let's join a new nucleotide to this very what? To this very DNA, so that you understand something. Now if I should join this very nucleotide to that very DNA, you see that what? There is OH at the bottom of that DNA, and we have OH also at the bottom of this very, or like at the top of this very nucleotide. So if you want to join them together, that means that what? There will be an elimination of what? Of water molecule, that's H2O. So you'll be left with what? With O. Now the same thing also would happen to what? To this very RNA. If you actually want to join them together, there will be an elimination of what? Of water. So that's just and this very reaction is called condensation reaction. So condensation will occur. Do you understand? So just note that you could just be asked. And everything you can see here is just what? Just a symbol, just to symbolize what I've actually explained in that DNA and RNA part. So these scientists made it possible for us to understand DNA very well. Do you understand? So from Shagaf's data, we have what? We have one ratio of purine to what? To pyrimidine. So let's just write that out. One purine is attached to what? To another pyrimidine. So that's what we can get from that very what? From that very data. One ratio of purine to what? To another one ratio of pyrimidine in our DNA. Now, after that, we have what? Rosalind Franklin. She actually got that what? She actually got her data through what? Through her X-ray crystallography. So you can just say S3. Let me just leave it like that. S3 something something. Now there is another guy, Paulins. He actually got what? This very data, but he got the DNA to be what? To be a three-stranded helix. Three-stranded helix, just like this. So that means he actually gave us this, that what? There should be what? There should be bases outside. So there will be bases here. There will be bases here. So the basis will be like this. The basis will be like this. Now would have what? Would have the what would I call it? Would have our what? Our phosphate group inside. And that can't be possible. So James Watson and Francis Crick, the what? They they actually came up with another structure of what? Of our DNA months after Paulins gave us this. So they came up with another what? Structure of DNA, and that is the accepted structure of DNA we have till today. That's what? That's a two-stranded helix. Do you understand? So where we have the base inside, the base are actually pairing inside the what? That very DNA. So we have the bases here, we have the bases here, do you understand? We have the bases, we have the bases here. Do you understand? So that's exactly the structure of DNA we still know till today. Now, just like the data we got from what? Watson and Crick, where we have our DNA to be a double-stranded helix. So let's just do that right now. Now, after we've done that, the next thing you should understand is what? Is the fact that what? That this very DNA, you have what? You have bases there. The bases are pairing actually with each other. And we will do that right now. So we'll pair these very bases now. Let's pair them. So there will be a base here. Let's call that one purine. Now, with that very purine, there will be another pyrimidine attached. Do you understand? Pyrimidine. We have, let's call the first one, let's call this very one pyrimidine, and let's call the second one I would use the red color for to be what? To be purine. So that's how they would pair with what? With each other. Do you understand? So that's just how they pair. And you can also see that what? You can also have that very structure here also. Then we would have what? We would have our pyrimidine also somehow. You understand? Now, the reason why I would like to I would like you to understand this is this: between these very what? Between these very structures, between this very structure, like here, between that purine and pyrimidine, we actually have a bond there called hydrogen bond. Do you understand? Now, this very hydrogen bond, it depends on the what? Of the purine and pyrimidine that are actually pairing. It could be two, and it could be what? It could be three. But I will talk about that. I'll talk about that shortly. Right now, I will talk about that shortly. But before I talk about that, let me quickly talk about this very DNA, that I was explained the other time. Now, this very DNA now, we have what? We have, just note that what? We draw a sequence of DNA from what? From 5 prime to what? To 3 prime. And I will explain that right now. You know, this very place, we have 1 prime here, we have 2 prime here, we have 3 prime here, we have what? We have 4 prime here, and we have what? 5 prime here. So that's where we go. We can actually see that 5 prime. Do you understand? Now, if you check this very place also, I'm sorry, I didn't put OH here. There should be OH there. Do you understand? And that is the difference between like now you should understand that the one of the difference between DNA and RNA is the what deoxyribose and ribose. In this very place, if there should be something here, if there should be OH here, that means what is a ribose. Do you understand? The ribose, that will be for what? RNA. If there's OH here, that will be for RNA. There's OH here also at this very carbon 2, that will be what? That will be RNA. That will change the sugar to ribose, and it will turn to what? RNA. But because we don't have something like that there, it signifies that what we're actually dealing with, what? With DNA. Do you understand? So now at this very OH, you realize that what? That's the carbon 3 actually there. Do you understand? Okay, let me just number it. We have 1 here, we have 2 here, 2 prime, 1 prime, and we have 3 prime. So that's why we actually build sequence from 5 prime to 3 prime. I didn't tell you the other time, I should have told you. You understand? Now, the second complementary strand, if you check this very strand, you see that what? If you check this very strand here, you see that what? It's just like a duplicate of this very strand here. But what I did is that what? I turned it upside down, and at the same time, I changed the position to the left. And the reason is that what? The reason is that in our DNA we have 2 strands, and these very strands, they are what? They are anti-parallel to each other. One is from one would be what? 5 prime to 3 prime, just like you can see here. Now this one, the second one also will be what? There will be 5 prime here, there will be 5 prime here, just like you can see. If I should count it, you see that what? The sequence will start from this very side. That's 1 prime, this is 2 prime, this is 3 prime, this is 4 prime, and I have 5 prime here. So that means that what? It's actually starting from 5 prime here, and if you check this very place, you see that what? At this very side, I didn't include it again. I'm sorry. There should be OH here. You understand? Now that very side, that's the 3 prime end. So we actually build sequence from 5 prime to 3 prime. Just understand that. You understand? We build sequence from 5 prime to 3 prime. Now, just like I did the other time, let me now try to do that again so that you understand it. I actually added a new nucleotide from what? Like on this very place to what? To build the sequence from 5 prime to what? To 3 prime. I added it to this very 3 prime end. Do you understand? So I can't actually add it here. Do you think I can add it here? I can't add it here. Do you understand? I can't add it at this very 5 prime. It is what? It's at this very what? 3 prime. So we actually build sequence from what? 5 prime to 3 prime. Now let's talk about the hydrogen bonding pattern of this very purine to pyrimidine, so that after that we'll be able to talk about the complementary strand of what? Of DNA. Just like I said earlier, through Shagaf's data, we can actually understand that what? It is 1-to-1 ratio of purine to what? To pyrimidine. So we actually pair a purine to what? To a pyrimidine. Now, example of purine, we have adenine, right? So we pair adenine A with what? We pair adenine A with another pyrimidine. So what's another pyrimidine that we pair A with? We pair it with what? We pair it with thymine. So that's it. What of the second one? The second purine, we have what? We have after adenine it is guanine, right? So we have guanine also. Guanine. So we pair this very purine with what? With another pyrimidine. What is that very pyrimidine? It is what? It is cytosine. So we pair guanine with what? With cytosine. So that's how we pair those bases in what? In a DNA. Do you understand? Now I want to, this is just like what you can see on the screen right now. You can actually see the structure of what? The first structure you can actually see here is the structure of what? Is the structure of what? Guanine. While the second structure you can actually see here is what? Is the structure of what? Is the structure of cytosine. So this very diagram would help us to understand base pairing in what? In these two structures, and we'll be able to what? Know the amount of carbon, the amount of hydrogen bonding between them. Now on this oxygen, you realize that what? There are 2 lone pairs on this very oxygen. So that shows that what is an acceptor of proton. Here, if we check this very H also, because it is what is an hydrogen on what on a nitrogen, that means what it will be what it will be a donor. You understand? Now, this very NH also, because it is an hydrogen, like it is a hydrogen on a nitrogen, should tell you that what it is a donor. If you check this very one also, this one also will be a donor. Now, this also would be what would be an acceptor. Would have this also to be what, because it's what it has two lone pairs, it will be what it will be an acceptor. You understand? So that means that we can actually pair these what these two bases, because what we have hydrogen on one side, we have like I said hydrogen, we have acceptor on one side, we have the donor at the other side. So we can actually pair them. You see, you can pair this acceptor and donor, you can pair this donor and acceptor, you can pair this donor and acceptor, making what making three between guanine and what and cytosine. You can just write that out. So it will be what anywhere you see that, that's actually three bonds. So we have G here, and we have what we have C here. You understand? So that's just that. So now let's talk about the second one. That is what that is the association of E and what and T. That's adenine and thymine. So this is our what? This is our adenine, and at this very side we have our what? We have our thymine. You understand? So let's quickly pair them together to understand just exactly what we did to what to guanine and what and cytosine. So this very place, you know that what it is a hydrogen on a nitrogen. So we have what we have these to be a what we have these to be a donor. This very O also, because it has two lone pairs, it's what it will be an acceptor. Do you understand? This also would be an acceptor. This also would be an okay. It won't be an acceptor. This very H would make it a what? Would make it a donor. So it will be a donor. How about this? This is what? This is actually a donor, and we also have this also to be what? To be a donor. So let's quickly pair them together. This would because these are donor and these are an acceptor, would have them to pair with like there will be an hydrogen bond there. This is also acceptor and donor, there will be an hydrogen bond there. But how about this donor and donor? There wouldn't be anything there, because what? Because donor and donor cannot interfere with each other. So do you understand? So we don't have any hydrogen bond here. So that shows that what we have only two hydrogen bonds in what? In the structure of what? Only two hydrogen bonds in the structure of adenine and what and thymine. So previously we got what three for what for guanine and cytosine. This one is what two. That's adenine and thymine. So just write that out, adenine and thymine. So note this because you could just be asked. Now let's come back to what to the covalent backbone of what of DNA. I was talking about this the other time that I would like to talk about the hydrogen bonding pattern of what of this very adenine and thymine and guanine and cytosine. I think you should understand that right now. So let me quickly give these. Let me quickly label this. Okay, let's make this A. Let the hydrogenous nitrogenous base there be adenine. The second one here, let it be what? Let it be guanine, and let this very one be what? Let it be thymine. You understand? Now, what will be the complementary base of this very A? The complementary base of A will be what? Just like I told you the other time, I said A would always pair with what? With T. Do you understand? So that means that what? That means that you would have T here. That is, if this very if this very purine is what is A, sure, it is very sure that this very purine will be what will be will be thymine. Do you understand? So that's just the scope. Now G, what of G? G would always pair with what? What I told you that time, I said G would always pair with what? With C. G would always pair with C. So GC and AT. Just try to understand that. GC. So, and what of T? T is A. We've already known that. Now let's pair them together. Let's pair them. This very A and T, we have what? We have two hydrogen bonds. So that will be 1 and 2. Just like I told you that time. Now, how about G and C? G and C would be what? Would be 3 hydrogen bonds. You know, we got it ourselves the other time. So that will be 1, 2, 2, 3. So we have 3 hydrogen bonds between what? Between G and C. How about C and A? That will be what? That will be 2 hydrogen bonds between T and what? And A. Now let's quickly talk about complementary strands, and we'll talk about what as well. We'll talk about the difference between DNA and RNA and some other things before we call it a day. Just like you know that we pair what? We pair G with what? We pair G with C. We pair T also with what? With A. So that's what we are going to use right now. This very 5' would what? Would be 3' here. Then we have A here, right? So that very A would be what? Would be T here. We have T here, right? So that one also will be A here. How about this very T? T will turn to A, C will turn to G. So let's write that there. A, G. This very one will turn to C. This also will turn to C. So let's write this here. C, C. How about this very C? It will turn to G. This very T will turn to A. So that will be what? That will be, so we would have what? We would have G, A. Now, so we would write the instead of this very 3', it will turn to what? It will turn to 5' here. 5'. And we are through. So that's the complementary strand of this very strand, I tell you. Now, what are the differences between what? Between DNA and what? And RNA. Let's start from what? From the structure of the sugar. Now, talking about this very nucleotide, you know, when I was talking about nucleotide, I said what? I said in our DNA, you would realize what? You realize the sugar to be what? To be deoxyribose. But when you're talking about RNA, you realize the sugar to be what? To be ribose. So that's the first difference between the DNA and RNA. So just write that. Deoxyribose. That's the sugar. And in RNA, we have what? We have ribose. Just that OH and OH we were talking about the other time. I tell you, it talks about the significant difference of what? Of DNA from RNA. Looking at DNA is stable throughout the lifetime of that very what? Of that very cell. But talking about RNA is what? Is unstable due to just OH and H. You see? Now let's talk about the second one. The second one is that what? If you look at it, you see that what? It's actually the nitrogenous bases that we were talking about the other time. We can find A here, that's adenine in DNA. You can find thymine in DNA. You can find what? You can find guanine in DNA. You can also see what we mean by what? Cytosine in DNA. Do you understand? But talking about RNA, you can find adenine. You can find guanine. You can find what? You can find cytosine. But the fourth one will be what? Will be uracil. Now, another one is that DNA is what? Is predominantly double-stranded. So just write that down. Predominantly double-stranded. Double what? Double-stranded. While talking about this very RNA is what? Is predominantly single stranded. So let's write that down also. We have RNA to be what? To be predominantly single stranded. Single stranded. And in addition, you've actually gotten it from what? From Watson and Crick the other time. They defined DNA to be what? To be a double-stranded helix. Counter to what Paulins said the other time, and